Well, hello everyone. My name is Shireen Shaban. I'm a laboratory director at the Molecular Genetics Lab here at ImaGenetics. Welcome to our series of the ImaGenetics community lectures. Today we will be talking about genetics and how it's affecting many aspects of our daily life. So while genetics is now at the forefront of medicine, the interest in heredity is very old. And perhaps it was triggered by people noticing the similarities of individuals within the same family, like identical twins or kids looking like their parents or siblings looking alike. But the modern science of genetics has started around 1800s with the efforts of like Mendel, Darwin with his theory of evolution, and then the journey started and is continuing till today. With major landmarks such as the discovery of the double helix of the DNA and the discovery of PCR, which is polymerase chain reaction, the method that we use in the lab to amplify DNA. And one of the most important turning points in the journey was the completion of the first human genome project, which enabled us to have a reference to use for future experiments and models of how we understand DNA and genetics in general. And just to give you an idea, this journey that started in the 1800s and continuing till today, it had harnessed over 12 Nobel Prizes. So how did genetics revolutionize medicine? Well, it had done so in many ways. Our understanding of genetic diseases has changed. Our ability to develop drugs, vaccines, to prevent diseases, to help in family planning, or to even treat some genetic diseases. So now our understanding of the structure of the DNA, we are able also to link certain genes to certain genetic disorders. We understand different types of mutations that can happen in genes and result in disease. We also can understand how genetic diseases are passed on, whether they appear in a form that we call sporadic, which is like the person could be the only one in his family having a disease, or it can be inherited, either in a Mendelian simple way, like when you hear of a person who has a recessive disease or a dominant disease, or it can be inherited in a complex form. And by complex, we mean it could be multiple genes that cause that disease by interacting together, or it could be interaction between genes and environment. Just having a gene mutated doesn't make the disease show up, but if you add some environmental factors, the disease might manifest in a patient. The technologies have also enabled us to better diagnose genetic diseases, and we do that by either sequencing, and sequencing is just simply reading the sequence, the nucleotide sequence along the DNA. We can either sequence a single gene, or we can sequence multiple sets of genes that we think can cause a disease, or we can sequence the coding part of the gene of the genome, what we call whole exome sequencing, or we can sequence the coding and non-coding parts of the genome, which we call whole genome sequencing. It's also very important for us when we are trying to diagnose a genetic disease to test the individual that has the disease, and in many cases to test other family members. And that helps us understand better how the disease is segregating in the family and make us implicate or a gene in the disease or not. And we have been enabled in the modern years by a technology called next generation sequencing, which is a simple, fast, and high throughput method that allows us to perform sequencing in a very rapid and efficient way. And together with the technology, there has been a dramatic drop in the prices of sequencing or the cost of sequencing. And that, of course, translates into easier and faster diagnosis. And just to give you an idea, the first human genome project, it took us 13 years to complete, and it cost about $5 billion. Now, with the new advances in the technology, we can get the same genome sequence in 2 days with the cost of a few $1,000s. And that naturally has interpreted into a surge in the number of publications, in the surge in the number of new disease genes identified, in the surge of a number of diseases that have been explained and categorized carefully. Genetics has also enabled the pharmaceutical industry by adding an ability to develop newer drugs, mainly using a technology called recombinant DNA. And in recombinant DNA, you can simply mix the DNA from two different organisms. And that had enabled us to make vaccines, to make drugs such as insulin or clotting factors, or some treatments for cancer and viruses. All of this would not have been possible without genetics. We can also prevent diseases either using the vaccines that we just created, as I mentioned, in the pharmaceutical industry, or by assessing the risk for diseases. And when we do that, we simply collect genetic information from individuals, their families, their ancestry. And that enables us to assess whether a person is susceptible to a certain disease. And when we are able to do that, it simply means that we can better counsel the patient. We can tell him about his own ability or his own chance of developing a disease and the chance of him passing on that disease to his children. So people can be informed about their own health and the health of their family members. Along that line, here at Imagenetics, we offer a test called Sanford Chip, which is a simple genetic screening test that provides two sets of information. Pharmacogenetics, which is simply a way to look at how your body metabolizes or processes medications. And the other set is your disease predisposition. We can assess whether you have a certain risk for cancer, for heart disease. And when you are informed of those, both your pharmacogenetic background and your disease predisposition, your physician can work with you to better guide your care. Genetics has also transformed the field of reproductive genetics and family planning. We can now perform carrier testing, which is simply testing parents for whether they carry mutations that can be passed on to their children. And then we can assess the risk for those kids in case they might develop a disease or not. We can also perform pre-implantation diagnosis in cases when in vitro fertilization is being done. We can test the embryos before they are being implanted, and we can remove those that can have a genetic disease and use only the ones that seem to not have any mutations. We can also perform prenatal diagnosis, which is a test during pregnancy by collecting samples from either the fluid that surrounds the baby or from the chorion, which is the attachment between the embryo and the mom. And we can test that material to see if the embryo can develop a genetic disease later on. We can also perform newborn screening. Newborn screening is a public health initiative that is conducted in most states. Simply by collecting a drop of blood from the newborn, those kids can be tested for early onset preventable diseases. Sometimes just providing some supplement can help save the life of a child, improve his quality of life later on, and maybe prevent the disease totally. One of the major advances and the one that carries the most potential is gene therapy. And by gene therapy, we mean the ability to manipulate the DNA, the genetic material, to either remove a mutated gene that causes a disease or to inactivate it, or in some cases even introduce a whole new gene into the system of a person in order to remove or treat or prevent a disease. Most recently, the FDA had approved one of those drugs for treatment of certain types of inherited vision loss or retinal degenerations. Sanford this year had awarded the very prestigious Lorraine Cross Award to two scientists, Dr. Jean Pennett and Dr. Catherine High from UPenn, for their groundbreaking work on retinal degenerations and gene therapy. They have made major advances, and I would encourage you to look up their work. And they have a lot of YouTube videos showing success stories and how the treatments they provided have changed the course of life of mostly children affected by those retinal degenerations. So it's quite promising field. So let's shift gears and move away from medicine to see how the genetic change other fields of our life or aspects of our life. And let's talk about animal and plant engineering. Genetic engineering in general means changing the DNA of an organism in order to produce new characteristics that we would like to see in that organism or that animal or plant. And the reason we would want to perform animal or plant engineering is that it carries the potential of improving human health, maybe increase food production, solve the problem of world hunger. In certain examples, we might even try to help the environment by decreasing the environmental impact of animal waste, or even improve the health of the animals we are working on. The only allowed animal engineering experiments so far by regulations in the country are recombinant DNA that allows us to make medicines or vaccines, and the transgenic mice that we use in the labs to model diseases and try to find treatments for them. There is still research going on. For example, we're trying to see if cows can produce milk for people who are lactose intolerant, or if the cows can produce milk that is similar to human breast milk, or some animals that are resistant to some diseases that are common in the animals, or as I mentioned, animals that can produce less phosphorus, or what we call environment-friendly animals. We also have engineered a lot of foods or plants, but there remains many questions about that field of animal and plant engineering. We're not quite sure if they have harmful health effects. We're not sure if we are manipulating the natural ecosystems and we might get bad results out of that. Also, if we are creating or engineering plants to be more resistant to herbicides, does that mean that farmers might use more herbicides? So in other words, we are harming the environment rather than helping it. And also, we are not quite sure if there's adequate government oversight. So although the field is growing, there are still remain to be concerns and questions to be answered. Genetics has also transformed the field of forensic science, mainly through DNA profiling, which is creating a certain sequence or profile for each one of us that can be used for legal and investigative purposes when needed. And to profile someone, you would need a DNA sample, and that DNA sample can come from blood, from saliva, from any samples that can be collected at a crime scene, for example, such as tissues under nails, semen, hairs, etc. And we perform, the most common test that's performed is called short tandem repeat testing, which is simply testing for certain markers. Each of those markers has multiple repeats that can differ from one individual to the other. So if you test multiple markers, each of them has multiple forms of repeats, we can create a unique profile for each individual, and then we can map that profile to any DNA profile that was identified in a crime scene, and hence we can link a victim or criminal to a crime scene. Currently, there is a system called CODIS, which is the Combined DNA Index System that the FBI uses to create such profiles, and it includes about 20 markers that have multiple repeats. And it creates a database where law enforcement can use that to go back to try to identify criminals if needed. We can also use mitochondrial DNA in cases where the nuclear DNA that we routinely use is not available. Mitochondrial DNA is more resilient and can stand worse conditions, and hence it can be an alternative if you can't find nuclear DNA. So the first case that used DNA as evidence to convict someone was the case of Timothy Wilson Spencer. He was accused of raping and killing 5 women in Virginia. In the year 1988, DNA evidence was used to convict him. At the same time, it was used to exonerate someone else who was convicted of killing one of those women. But of course, when Spencer was convicted, naturally the other individual was exonerated. Another aspect of forensic genetics is animal forensic genetics. We can use genetics to identify animals that could be victims of crimes, such as being stolen, or in case of identifying remains of a lost animal, or in convicting animals in case of attacks. If an animal attacked another animal or an individual or caused property damage, we can use that animal's DNA to prove if that animal was actually the perpetrator. We can also use animals. They could be witnesses in crime scenes. So if you find, for example, DNA from a pet of an individual in a crime scene, that means that that individual might have been at the scene at the time of committing the crime, and that can be weighed in court to see if that exonerates or it incriminates an individual. We can also use forensic genetics for animals in an attempt to stop the practice of dog fighting. There is an effort now to create a canine code system, such as the FBI code system for human beings, in order to track individuals who perform such practice and try to prevent that. We can also save wildlife by performing forensic testing on animals to prevent inbreeding, which is mating of closely related animals, which can put the animals at the risk of recessive diseases. Or we can use it to eliminate the crimes of international animal trafficking or illegal hunting. Another very effective way genetics has been used is its ability to help unite families and find the lost. A recent movie that has been awarded a lot of awards, including the Sundance Film Festival, showed the story of three triplets that were separated at birth and given to three different families living under three different conditions. And it turned out that that was part of a very unethical experiment that separated those twins, either those triplets or other examples of twins, in order to test the effect of environment on individuals, given that those are twins, so we assume that they share the same genetic material. So any changes that happen to them over their lifetime would be attributed to the environment. Using genetics, those individuals finally managed to find each other and be reunited. It's a tragic story, and out of it came another nicer story of a lady who watched the movie and discovered that she was adopted through the same agency that mediated the adoption of the other three individuals in the movie, and she tracked that adoption agency and could actually find her sister, another twin sister that she never knew about. So DNA can actually help us reunite families. I think many of you have heard of the tragedy at the border nowadays with the separation of kids from parents, those who are seeking asylum at the southern borders, have been separated from their children, but DNA is being used to try to reunite those families with their kids again. Also, DNA can be used to identify remains in tragedies like a genocide or a civil war, or sometimes horrible terroristic acts such as 9/11. You can use DNA to identify the remains of the victims in such cases and link them to their relatives, and hence identify their names and give the remains back to their families. Also, another aspect that genetics has touched and affected is our understanding of the human origin. Archaeology is the science of studying human history and prehistory. And archaeologists actually use DNA by analyzing samples from very ancient times, human remains, or whatever they find as remains in caves or wherever they are investigating remains. And they can help define or create a model of our history. Many of you might have heard of the expression out of Africa. Through archaeology and through DNA testing, it had been proved that all races, all populations, African and non-African, had originated actually in Africa. And then there were major migrations into Asia and Europe and other continents later on. We can also use DNA to identify mummies and learn more about ancient civilizations and human characteristics at the time of such existence. One very interesting aspect is also the ability to use genetics in space science. NASA has been very fortunate to have two astronauts who were identical twins, and they performed that wonderful experience of keeping one of the identical twins who share the same genetic material. They kept one here on Earth and sent the other into the outer space for 6 months. And then when he returned, they tried to compare the genetic material of both individuals. And interestingly, they found over 7% change in gene expression, which is quite striking. We all might suffer differences in gene expression in cases such as scuba diving or individuals who climb mountains, but that the percentage was quite high and was worth investigating. We are still trying to understand what those changes mean and if they would be permanent or could be reversed as the person returns to Earth. But it was quite interesting to document such change. We are also investigating whether plants and animals can tolerate the zero gravity in outer space, or what we need to do to be able to grow plants, and that in the space, which means that in the future we might have farms in the outer space to compensate for any lands that we lose here on Earth. Lastly, and most fun, is genetics can help you learn more about your animals. There are many commercially available tests that can help you take a simple swab from your animal, chick swab, and test your animal to understand more about their background, with which breed they belong to. And it usually shows you if they are a purebred or like a pure breed or a mixture of multiple breeds together. It's quite fun. And lastly, also, we know that in horses that are being used in horse racing, genetics has been used for a long time to confirm their parentage. You know, some of those horses are worth millions of dollars given that they are of a certain parentage. So we can use genetics to confirm that. Up until 2012, modified animals or modified horses were not allowed, genetically modified animals were not allowed to compete, but that had changed, although the debate continues about whether this should be allowed or not. Well, thank you everyone, and I hope you found our talk today useful, and I hope I have shown you that how genetics has changed many aspects of our life, from medicine to animal plant engineering, science, space, forensics, and many more fields that genetics is actually affecting or will affect in the future.